It's a pleasure for me to introduce the next speaker, that is Giovanni Melioli. It's my pleasure because he's not only a dear friend and also a mentor. And Giovanni is a well-known scientist, actually, in, I would say, at the interface between microbiology and respiratory disease. And as a matter of fact, he's going to tell us about microbiome in inflammatory respiratory disease. Gianni, please.
Thank you very much. Thank you for the invitation. And I'm really excited because this is the first time I speak about the airway microbiota. And so being the first time, everything is much more difficult. Okay, let's move just for the beginning from what it is the approach of classical microbiology. There are pros that are characterized by the identification of bacteria that have an intact proliferative potential. So it means they are able to produce colonies. And using this proliferative potential, we can analyze the antibiotic susceptibility. The contrary is that we can detect only microbes that are able to survive and growth. So there is insufficient of no detection of anaerobic, and we have no idea, using classic microbiology, of the functional subsets of the respiratory tract. In addition, saprophytic flora, I have an idea of what is a saprophytic flora in the picture of the agar plates on the bottom. Saprophytic flora was never considered relevant, but now we know that the things are completely different. Which are the microbes of the respiratory tract? Well, we have three districts. One district is the oral and the oropharynx, where we have a resident microbiota. It means that we have microbes in our throat. We have upper respiratory airways. Always the microbiota is resident, and lower respiratory airways that are, for definition, considered as sterile. But under disease, they can be infected by microbes. And you can see that many microbes belonging to the upper respiratory tract are also found during disease in the lower. But what is actually relevant is that if we look at the real-life practice, we observe that oropharyngeal swabs, as well as sputum analysis from a bacteriology point of view, is generally largely negative, 85% versus 7-8%. It means that using classic microbiological methods, we have big difficulties in identifying microbes in the respiratory tract. There is a very long story of love between prokaryotics and eukaryotics. Prokaryotics appeared 3 billion years ago, eukaryotics 600 million years ago. So it means that we spend about 600 million of our life as eukaryotics in a strict convivence, but was extremely fruitful convivence with microbes. And this should be kept in strict consideration. The term microbiota appears in 1986. So we are speaking about a science that has almost 30 years, and it's a new science in particular for the application at the clinical level. Some definition. What is microbiome? Microbiome consider all the microbes in an organism, or, from a molecular point of view, a collection of all the genes present in microbial cells. Just to give you an idea, our genus that are Homo has about 30,000 genes. All the microbes of our gut microbiota has about 300,000 genes. So microbes of the gut offer us a lot of new properties, of interesting capacities that we do not have for genetical reason. The microbiota, in contrast, is the description of the bacteria in a single organ. And, for example, we can speak about cutaneous microbiota, oral microbiota, respiratory, gut, etc., etc., etc. And what it is relevant is that the microbiota analysis combines microbiology, genomics, bioinformatics, ecology, system biology, all sciences that are focused in elucidating the contribute made by microbes in functioning of the organism. Which are the functions of the respiratory microbiota? Well, a barrier function, modulation of the immune response, metabolic function, maintenance of homeostasis, influence on the inflammatory response, protection against pathogens, and regulation of mucus production. It means that we have a lot of different functions associated to the microbiota. But also, it has been clearly shown that the microbiota may have a potential role in disease. So we have a balance between the healthy microbiota and the microbiota of sick. And which are the factors that influence the microbiota? Gender, for example, antibiotics, chronic disease, diet and nutrition, genetic factors, and so on, up to smoking and vaccination and so on. So the number of factors that are able to interfere with the homeostasis or respiratory microbiota are many. If we want to study the microbiota, we have two approaches: a classic microbiological method and the molecular approach. The classic microbiological method starts with a collection of biological samples that must maintain living the microbes, because if we kill the microbes during the pre-analytical phase, we cannot observe anything. Then we have to plate on agar place, detection of colonies, identification of the microbes, and evaluation of the susceptibility to antibiotics. On the contrary, if we move to the molecular biology approach that is based on the next generation sequencing, we have the collection of biological samples using media suitable to maintain DNA. Then we have an extraction of DNA, an expansion of the 16S genes using the PCR, next generation sequencing, and finally, informatics identification of different taxa of the microbes that are present. The method is complex, so the title that Guido suggested me, lab skills, yes, we need a very strong lab skills made by expert biologists. We spend one day and a half for preparing one microbiota, 1 day for the sequencing, and 6 hours for interpretation. So when we speak about a single assay, we speak about a lot of work. In addition, we have the second phase that is represented by the interpretation. What we observe from our instruments are something like that, and we have to move from this sequence to the identification of the microbe. So this is a very strong, very big bioinformatic work that should be done in order to transform the data that we obtain from the sequencing procedure to something that is useful, for example, in clinics. In December 2023, together with Guido, Fulvio, and very few others, we decided, and with the collaboration of Bruschettini, that is a company in Genoa that are all friends, we started analyzing the human respiratory microbiota. And for that, we collected 11 donors, 9 patients with acute respiratory infection treated with antibiotic, and 6 patients with chronic respiratory disease. And in all these patients, we evaluated the absolute number of species detected, the parameters of biodiversity, the fraction of data of taxa that can be identified, and finally the functional parameters. So we decide to go deeply into the analysis in order to have an accurate picture of what the respiratory microbiota is trying to tell us. And these are very, very preliminary data. So this is the reason why I'm from one side excited, but from the other side a little bit worried. First of all, the biodiversity. We have two parameters. A parameter that is named just biodiversity, and the other is a Shannon test. Biodiversity is 433 in healthy donors, but is 76 in antibiotic patient and 121 in severe disease chronic patient. And the Shannon index is 223 versus 145, 127. What it means? It means that if we compare healthy donors with people that has a respiratory disease, independently from the fact that it is acute and treated and from the fact that it is chronic, we observe differences. And why these biodiversity indexes are so relevant? Because high biodiversity is associated with healthy airways. On the contrary, low biodiversity is linked to pathological condition. This is a general rule. It works for the gut, for the nose, for the airways. But in the present situation represented by respiratory airways, this is a very important and also very clear data. Let's look on the quantitative analysis. I don't want to comment this one, but to make short a long story, these are healthy donors. We have 100 different microbes. This is an antibiotic patient, 2 microbes, and this is a severe disease patient, 20 microbes. So what it means? It means that if we look at the number of microbes that are available in the swabs, in the materials that we can collect, we observe very big differences. And the big difference are about 95% reduction for donors, for antibiotic patient, and 80% reduction for a severe disease patient. Considering that biodiversity is extremely relevant for the maintaining of homeostasis of the respiratory tract, this is a very relevant point. Qualitative analysis. Let's compare donors with acute patient treated with antibiotics. If the population were identical, we should have a line with no histograms. As you can see, we have a very, very big difference. And in certain situations, the increase is 225,000%. It means from nothing to a very, very large population of microbes that are not so common when we observe the results of clinical classic microbiology. So, what can we observe? A marked reduction of the gram-positive saprophytes, reduction of the defense, emergence of pathogenic gram-negative bacteria, and an expected increase of actinobacteria. And look at these microbes. These are very far from the microbes that are usually we consider as related to a disease. Let's look the difference between Let's look the difference against the donors and severe chronic patient. Something similar with small difference. For example, the emergence in this patient of gram-positive bacteria such as the Staph A that are not very difficult. That are not so common in the normal respiratory tract, but also, again, the expansion of rare bacteria, such as actinobacteria, that we already observed in patients treated with antibiotics. What it means? It means that if we reduce the saprophytic population in the respiratory tract, we observe the emergence of something that is not as expected, as is not so safe. If we decide to work a little bit with the functional capabilities, we can detect that different characteristics can be studied. For example, which are the microbes that are present, if we have or we have not antipathogenic activities, and which are the bacterial function. Something of these were already described before. And this is a complex picture, but to make short another long story, if we look at healthy donors, we have only 6% reds. It means 6% results that are not as expected, against 43% reds in severe chronic severe disease and 75 reds in acute disease with antibiotics. So we have a worsening, a significant worsening of the microbiological situation, moving from healthy donors to other disease. And if we want to use using machine learning, so artificial intelligence is involved. If we want to use machine learning to have a sort of control of our diagnosis, we can observe that also machine learning, using this approach, is able to identify very few unexpected results. A severe patient that was classified in healthy donors, and a sort of mixtures on the boundary between antibiotic patient and a severe chronic disease. So these two approaches, one supporting the other, meaning that if we use the functional characteristics to describe what are the activities of the microbes of the respiratory tract, we have suitable results. The question is it possible from this data to develop a report for clinical use? And this was my work. You know that I'm a microbiologist, but also a clinical pathologist. So, for example, this is the report that is made in our lab, and in using machine learning and algorithms of artificial intelligence, it is very easy to describe this situation as a balanced respiratory microbiome. On the contrary, here we have a chronic disease patient with an absence of imbalance, absence of balance or an imbalance of in the respiratory microbiome, and the same in patients using antibiotics. You see all the red dots indicating that the results are out of the reference ranges that we use to understand what we are doing. And also, if we work on the frequency of different taxa using such an histogram, it is very easy to detect that the proteobacteria, for example, are extremely high in the presence of unexpected low results, and other results, other taxa, other phyla, are small, are lower in the presence of a higher expected result. And the same is also valid if we looked on genus. You observe that, for example, Escherichia and Shigella that are virtually absent in a healthy donor microbiota is extremely high, reaching the 35% of the whole microbiota in this kind of patients. In addition, if you look at the species, we can also have something. For example, Veillonella or Escherichia coli are higher than expected, but also microbes that we usually observe in clinical microbiology are extremely rare. So in classic clinical microbiology, we add a selection that is not suitable to represent the real situation of the respiratory tract. Conclusion. We have basic evidences that classic microbes observed in clinical microbiology are represented in virtually insignificant fractions in the pathological samples. And only Klebsiella pneumoniae and Escherichia coli are barely increased in these patients. This is different from what we observe. From a quantitative level, that we can find that in healthy donors we observe a great number of microbes and a great heterogeneity, while in patients we have a low or a very low expression of the biodiversity as measured using molecular biology approach. And also at qualitative level, we have, for example, that actinobacteria and veillonella are strongly increased in patients treated with antibiotics. And this is not what we want giving antibiotics. And also, Staphylococcus epidermidis and other microbes belonging to the actinobacteria are strongly represented. But much more important, in all pathological samples, the bacteria that are characteristic of normal respiratory microbiota are lost. This means that we are losing all the functions that are expected to be offered, given us by the microbe. Also, by using this approach, so having a picture of the functional characteristic, we have a clear demonstration that we are observing completely different situations. What can we do? This is the approach that is used for the gut microbiota. First of all, working on the lifestyle, on food, prebiotics, probiotics, antibiotics, up to biological therapy and microbiota transplant. Similarly, we can think that also for the respiratory microbiota, we can work on the lifestyle, working on smoking and pollution, on the food, considering that the gut-lung axis is extremely relevant, working on probiotics. It has been shown that Lactobacillus bifidobacterium and Streptococcus salivarius are able to improve the quality of the microbiota, using prebiotics such as vaccine of any kind and also bacterial lysate, but also, if needed, working with really targeted antibiotics. And finally, and this is still in progress, biological therapy and microbiological transplant that are the future probably in this field, but at present are not something that is real evident. And this is the group that worked with me. I want to thank all the doctors belonging to the group of Fulvio Braido, because they helped me a lot, the colleagues of the microbiology of my lab, the group of molecular biologists, the bioinformatics, and the people that helped me in discussing in this 9 months of work, because as I told you, this is new for virtually everyone, and so is much more complex than what we expected before. Thank you very much.